Okay. So to reiterate, all the gene lists that we calculated, those 7 gene lists, this is how you're going to compare them. So you're going to download your gene list just like we did. Remember, you're going to have to figure out whether it's Illumina or Affymetrix. If you have any questions, go to the actual accession, find the accession number, go to Array Express or Geo and find out what array they used. Okay? What time is it? Okay, we got time. All right. So once you've done that and you've downloaded your gene list, this is how you're going to compare them. This is how I found my master list. Is you're going to go, if you look on this project manager, you'll go down to this comparison analysis. And if you have some gene lists, actually, I want to go down here. So I'm going to hit New Comparison Analysis. Okay. Now I'm going to go where my gene lists are, which are here. Yeah, we've done a lot of stuff. Yep. So here's what I'm going to do. I'm going to take these gene lists and I'm going to start adding them. There's Condor, there's me. There's me again. I think that was Mario's list. We're going to have to check your list, Mario. There's Pre-Medi, KT's original list, G-Love. Am I missing anybody? Yeah, where's yours? Oh, yours is down here. Actually, you had a pretty decent list. I'm going to move yours up a little bit. Yeah. All right. So 1, 2, 3, 4, 5, 6, 7. Again, these are 7 lists from 7 different lung cancer studies that looked at lung cancer stage. We took the top 1,000. Now we're going to compare the results between these. And this is some heavy duty bioinformatics here. So we're going to view comparison. And we've been kind of playing around with this. I know people have been in and out, so I want to kind of review this. All right. As easy as that. If you want to save before you get out of this, I mean, you can do these comparison analyses all the time, which is really, awesome. But if you want to save it, before you close it down, you have to hit save and exit. And then you have to name it. And it will basically save. See down here? I did a previous comparison just using 4 data sets. But it'll save in this window here. I'm pointing at my screen like you guys can see. Right there. Don't you see it? Follow the arrow. The arrow knows. So again, we can look at this canonical pathway here. This Fc epsilon Ri signaling, I'm going to tell you, you can look it up. And that's what I really want you to do is kind of Google some of these things. That what you're going to find, and we've been talking about this, is that you probably have already had cancer at some point in your life. It's just that your immune system took care of it, right? Here is the pathway. This is probably how it hides from the immune system. So we can look at some of these. Let's look at this one. I think it's opening. Come on. Oh, there it is. Yeah. So you got rack, you got these things coming down. So this would have been Condor's study. This was Mario's, I believe. Mario, I think the data set's kind of weird because you're actually showing an increase. So we can click here. Basically, what's going to happen is all of these things, remember, this is all just probabilities. All of these studies kind of pick this one out, except this one, as being overrepresented. When we click on each individual, so there's yours, BT, which had the strongest inhibition, you can see some of the same things lighten up. Isn't this cool? I think this is so awesome. But again, you can go down here, and if you know anything about lung cancer, and you will by the time you get done with this, because I want you to kind of look some of these things up, all these things make sense. mTOR signaling is huge in lung cancer. It's a signaling pathway for growth. And these lung cancer cells are exploiting it. And you can see they're using very specific signaling avenues. I don't like Rho, I don't like PKC, but I love RAC. I'm going to go through here. You can look at some of these other, oops, no, I hit the wrong one. There's a synapto, lots and lots of cell division stuff. We can look at diseases and functions. Again, this is what I want you to do. Look at the top one. What does that say? Can anybody read that?
Proliferation of cells.
Proliferation of cells. What does that sound like? Cancer. That's cancer in its simplest form. Proliferation of cells. And if we look here, we can actually see some of these things. Look at cancer is the second one. Look at, it's way in there, guys. This is so cool.
That is one gene.
No, this is a function, and all of these things that it's tied to on the outside. Let me see if I can.
I don't think that was just one gene.
So everything here was in our gene list. And they're all tied in some way or form to proliferation of cells. If it has an orange line, that means if it goes up, it promotes proliferation. If it has a blue line, if it goes down, then it means it promotes it as well. But yeah, this is basically, this is the nuts and bolts of lung cancer. We can look at some of these others. Cancer. Look at that one. Look at all these genes. Like I said, guys, there's a gold mine in here. The cures for cancer are in here. This is basically the language these cells, these tumors are speaking. You can think of this as Latin or, ancient Phoenician. It's in a language, really, and it's your job to figure out what it's saying. Right now, it's just shouting at me, cancer, I'm growing. I'm growing. I don't like dying. You can go through here, cell viability. All of these things kind of make sense. And then you can look at some of the things that actually decrease here. Microtubule dynamics. So remember, microtubules are like the skeleton in those cells. If it's going to divide, it's got to break down that skeleton system. And that's exactly what it's showing right here. Tox functions. Let's look at some of these. Top one, liver cancer, carcinoma, and it's all up. Basically, what I'm showing you now, the fact that this doesn't look like a Jimi Hendrix poster or some tie-dye shirt, is showing that what we did worked. That we expected to see a lot of cell growth in these cells because I just want to, remind you what we did is we used mathematics to take a blind knife and cut off just a little bit of data on each one of those studies. Then from that little bits, we found out what was the common bit, and now we're analyzing it. And this is telling us all kinds of stuff. Honestly, this is kind of like the wisdom of crowd experiment we did. Really. It's like, what do all these different studies say? If I take the average of what they say, it's probably more accurate than any individual study. And that's exactly what we did with this exercise here. What's some other ones? But again, I want you to kind of go through here. Upstream analysis. This is awesome. Come on. We got MYC is one of the highest ones. Again, what I want you to do, these are individual genes. I want you to go through and actually look some of these up. So the top upstream regulator is MYC. Here's what we can do. Let's go to our YouTube. Let's go MYC, MYC, lung cancer. Just Googling. MYC is metastasis gene for non-small cell lung cancer. This stuff works. The cells are trying to tell you something. They're yelling at you. Hey, these guys are making me do stuff. And we can see these. CSF2 is another one. But you can see the type of agreement here, that if you see a bunch of blue, it tends to be carried through all the different other studies. All these are more predicted to be more active, inhibited. And again, this is a huge tumor suppressor. But this is what I want you to do is, remember, we're going to find we want a common pathway. Actually, no, we can't do the networks on this one. What I want you to do is basically, start putting some diagrams together. I want you to use this comparison to give you an idea what might be important and what it's doing in each individual one. And then, is your data set